Hi, everyone. Welcome to the Lymphoma Research Foundation's webinar on highlights from the American Society of Hematology, or ASH, 2024 annual meeting. I'm Hope Avalone, and I'm the Senior Manager of Patient Education Programs at the Lymphoma Research Foundation. During today's webinar, you'll hear from a lymphoma specialist, and you'll have an opportunity to ask questions. You can ask those questions at any time in the Q&A box at the bottom of your screen, and we will address as many as we can after the presentation. And as a reminder, this webinar is being recorded. At the end of the program, you'll be prompted to complete a program evaluation. We do ask that you take the time to complete this evaluation, as we like to incorporate your feedback into future programming. But for now, let's get started. Thank you again to each of you for taking the time to join us on today's webinar. At the Lymphoma Research Foundation, our mission is to realize the promise of science to eradicate lymphoma and to serve the community touched by this disease. All of our work is led by our Scientific Advisory Board, which is a group of 45 leading lymphoma experts from around the country. And in addition to today's program, the Foundation has a variety of other programs and services available for you. We offer both in-person and virtual programs, so you can continue learning about the latest updates throughout the year. To view and register for any of our upcoming programs, you can visit lymphoma.org/programs. And in addition, the Foundation offers a wide range of support services. First, our helpline, which serves to complement our patient education programs, is a resource for both patients and care partners. The helpline has master's level trained staff who provide individualized information on all types of lymphomas, as well as information on treatment options, clinical trial navigation, and connections to resources such as financial assistance. We also offer a peer support program called the Lymphoma Support Network. And through the Lymphoma Support Network, we can connect patients and care partners with others who have been through similar experiences for emotional support. And the Foundation also offers a wide range of digital resources. Comprehensive disease guides and fact sheets can be either ordered or downloaded on our website, lymphoma.org, where you'll also find disease and treatment-specific videos. We also have a mobile app called Focus on Lymphoma, and this app provides helpful disease content as well as unique tools to help patients better manage their lymphoma. The app is available free of charge in the Apple App Store and in Google Play as well. I would also like to take a brief moment to thank our program supporters. Thanks to their generous support, we're able to host programs like today's webinar. But for now, we have a wonderful program planned for you today, and I'm honored to introduce our speaker, Dr. Anne Lacasse. Dr. Lacasse is an associate professor of medicine and a lymphoma specialist at Dana-Farber Cancer Institute, where she is the director of the Dana-Farber Fellowship in Hematology and Oncology. She is currently the chair of the Lymphoma Research Foundation Scientific Advisory Board. Dr. Lacasse is also a former recipient of the Foundation's Career Development Award, and she has served as a member of the Foundation's Scientific Advisory Board since 2012. We are very pleased to have her on this program today. So without further ado, Dr. Lacasse, I'll turn the talk over to you.
Thank you so much, Hope. I'm going to share some slides here. And I really appreciate everyone joining this afternoon. I think this was a big year in lymphoma at the ASH meeting, and today I'll be focusing on B-cell non-Hodgkin lymphoma. So these are my disclosures, none of which have any impact on what I'm going to say. So this is our agenda. I'm going to give a brief overview of B-cell lymphomas. Many of you may already have a good understanding of these diseases, but just in case there are those of you out there who are new to lymphoma, just a short overview, talk a little bit about the role of what is a clinical trial and what is the role of a clinical trial, and then focus on studies presented at ASH in 4 different diseases: diffuse large B-cell lymphoma, follicular lymphoma, mantle cell, and chronic lymphocytic leukemia, small lymphocytic lymphoma. And then we'll have hopefully some time for some questions.
Dr. Lakees, can you try unsharing and resharing? I think we can't see your slides advancing.
Okay, let me try it again.
Perfect.
Do you see it switch now?
Yes, much better.
Okay,
Thank you.
great. All right. So lymphoma is complicated. There are many different subtypes, and I'm sort of a lumper when I think about these. But the definition of lymphoma is that it is a cancer of the lymphocytes. These are part of your normal immune system. They arise from bone marrow stem cells, and then they go out to live in the lymphoid tissues, which are the lymph nodes, the spleen, and the bone marrow. Sorry for the typo there. But these lymphocytes can really hone to any area in the body. And the way that we classify lymphomas is based on getting a biopsy and having our pathology colleagues take a look and tell us what the disease is. And factors that they use to make that evaluation include: what do the individual cells look like? What is the pattern of those cells? And then they do some fancier tests called immunohistochemical stains and other testing to really come back with us and tell us what kind of lymphoma we're looking at. So broadly, we divide lymphoma into non-Hodgkin lymphoma and Hodgkin lymphoma. Hodgkin lymphoma is much less common, only about 8,500 new cases a year in the US. Tends to affect young adults, though we see it in all age ranges. It was described first, so all other cancers involving lymphocytes are lumped under non-Hodgkin lymphoma, of which there are more than 70 different subtypes, and much more common, about 85,000 new cases a year. And within non-Hodgkin lymphoma, we divide them into B-cell subtypes of non-Hodgkin lymphoma and T or NK cell lymphomas. So when we broadly think, there are many different subtypes, but I think the easiest way to sort of think about them is: how do they behave in patients? So if you look at this chart here, so for people who, if you weren't treated, how long would you survive with the lymphoma? What is the goal of our initial treatment, and what's an example of that lymphoma? So indolent means slow growing. So patients who have slow growing forms of non-Hodgkin lymphoma, for many patients, even if you didn't treat them, they would survive for years. The goal of our initial treatment, we have said for many years, is that it's to control the disease and put it in remission, but that we would typically expect that it would recur over time. I think that's an open question in this day and age, given some of our newer therapies, and we're seeing some patients who may have very long lasting remissions, such that their disease may not recur in the future. And sort of the prototypical slow-growing non-Hodgkin lymphoma is follicular lymphoma. Aggressive lymphomas, these progress more rapidly, and if we didn't treat them, patients would likely survive for months. Obviously, we do treat them, and the goal of our initial treatment is to try to eradicate or cure the lymphoma with the initial therapy. And the most common subtype of aggressive non-Hodgkin lymphoma is diffuse large B-cell lymphoma. Then the highly aggressive, these are lymphomas that grow extremely rapidly over weeks, sort of in front of your eyes. These are also potentially curable with standard chemoimmunotherapy. And the example here is Burkitt lymphoma, which is a rare subtype of non-Hodgkin lymphoma. So in terms of what are some of the other indolent non-Hodgkin lymphomas that we see, B-cell chronic lymphocytic leukemia or small lymphocytic lymphoma is a subtype of slow-growing non-Hodgkin lymphoma. The definition can be a little bit confusing. If we see more than 5,000 tumor cells in the blood, we call that chronic lymphocytic leukemia, emia meaning the cells are in the blood. If we have the same disease, but fewer than 5,000 of those tumor cells in the blood with involvement of lymph nodes or the spleen, we call that small lymphocytic lymphoma. But we really kind of treat those in the same fashion. Lymphoplasmacytic lymphoma and hairy cell leukemia are 2 rare subtypes of indolent non-Hodgkin lymphoma. I mentioned that follicular lymphoma is the most common indolent lymphoma we see. And grade means, what do the cells look like under the microscope? Are they small or are they larger? The larger ones imply that the disease is growing more rapidly, and we sometimes treat that differently. I would say here, grade 1, 2, and 3A, it's probably too much information for the context here, but these are diseases that we see commonly. Marginal zone lymphoma, there are 3 subtypes: nodal, extranodal, sometimes called MALT, mucosal associated lymphoid tissue, and splenic are indolent B-cell lymphomas. And then I put an asterisk next to mantle cell lymphoma because it has really quite a variable clinical behavior. It can behave like a slow-growing non-Hodgkin lymphoma, or there are some subsets that can be more aggressive, like an aggressive non-Hodgkin lymphoma. There are 2 indolent, these are 2 examples of indolent T-cell lymphomas. One is what's called T-cell large granular lymphocyte leukemia, and mycosis fungoides, which is a T-cell lymphoma involving the skin. Here are a couple examples of aggressive lymphomas. As I mentioned, diffuse large B-cell lymphoma is our most common one. Grade 3B follicular lymphoma, we treat just like diffuse large B-cell lymphoma. And then the T-cell lymphomas tend to be aggressive, and there are many different subtypes. I've just listed a couple different ones here, but we consider those aggressive. And then the highly aggressive lymphomas, I mentioned Burkitt lymphoma, another rare disease called adult T-cell lymphoma leukemia, which is a disease that we don't see so much in the US, but is a very fast-growing lymphoma. So I thought I'd spend a moment and talk about clinical trials. As many of you may know, this is where we test new treatments, and there are phases of clinical trials. The first phase of a clinical trial is based in the laboratory, where our scientist colleagues develop drugs that they think may be effective in lymphoma based on their biology and what we call preclinical evidence. If we see something that looks encouraging, we take that to a phase 1 study. The goal of a phase 1 study is to understand the safety and tolerability of that particular drug, and to figure out what is the appropriate drug dose that we want to move forward into subsequent trials. Phase 2 trials, and many of the studies that I'm going to show you today are phase 2 trials. These are studies where generally all of the patients receive the same therapy. And the goal of this is to treat a broader number of patients with the same treatment and really get a sense of how well the treatment works and what is the side effect profile. And generally, these studies have fewer than 100 participants, but there are exceptions. Phase 3 studies are studies that are often submitted to the FDA to get approval of a particular drug. These are often randomized, meaning patients will receive either the standard treatment or our new treatment. And we have to believe, if we're doing a phase 3 study, that this new treatment is going to be better than our standard treatment. And that's how we can justify doing a randomized trial. These are usually much larger trials, often 100 patients or more, depending upon how the trial is designed. And then phase 4 is really collecting data once a drug has been approved to make sure that we haven't missed any safety signals that may have not emerged in these prior phases, because we're not treating a large number of patients in the context of the phase 1 and 2 and 3 studies. So I thought I would start with what I'm going to do here is to present a select number of studies from the meeting that I think were interesting and important, and divide them up according to disease. These are an overview because we have a lot of particular studies to kind of get through, but welcome any questions at the end. So again, diffuse large B-cell lymphoma, this is the most common lymphoma subtype that we see in adults. It's about 25 to 30% of cases. It typically can affect any age, but the median age is around 65, and there's a slight predominance of males who develop this disease. And the name diffuse large B-cell lymphoma basically reflects the way it looks under the microscope. Diffuse means the cells are kind of spread apart. These are large cells, and they're B cells. So that's where the name came from. So chemotherapy in diffuse large B-cell lymphoma was really a sort of life-changing in oncology in general. Because prior to that time, we really didn't have great treatments prior to the 1970s. And in the 1970s, this combination of CHOP chemotherapy, which is a 4-drug chemotherapy regimen, was introduced. In the 80s and the 90s, there were new chemotherapy regimens that were devised, basically intensifying CHOP by adding additional drugs or giving them more frequently to try to overcome any resistance. And those studies, those were phase 2 studies where each study enrolled, all the patients got the same treatment. Those looked good, but then they did a randomized study and compared these more intensive regimens to CHOP. CHOP was just as good and less toxic. So CHOP really remained the standard of care in diffuse large B-cell lymphoma until 2002, when a study showed that adding the CD20-directed monoclonal antibody, the first targeted immunotherapy agent in lymphoma, to CHOP was better than treating with CHOP alone. And then for the next, about 20 years, there were many attempts again to improve on R-CHOP, giving it in a more intensive fashion or adding novel chemotherapy drugs to R-CHOP. And again, none of those studies actually showed a benefit overall. And that was until 2021 when this study added a drug called polatuzumab to chemotherapy, R-CHP, replacing the O drug or vincristine, because of both drugs have similar toxicity in terms of numbness and tingling, and comparing that back to R-CHOP chemotherapy. So at ASH this past year, the POLARIX study, which compared polatuzumab plus R-CHP chemotherapy to standard R-CHOP chemotherapy, was presented the 5-year analysis of this study. So this is how polatuzumab works, and this is a class of drugs called antibody drug conjugates that are used in many different types of cancer now. And basically, you have this antibody or protein here, and to it is linked to chemotherapy drugs. So the idea is that this drug binds to the surface of the tumor cell and delivers the chemotherapy directly into the tumor cell, thereby hopefully giving better cell death of that tumor cell while sparing the side effects of giving the chemotherapy just by vein, where they may get delivered all over the body. So this was the POLARIX design, and some of these studies are a little bit complicated, but basically tells you that this took adult patients with diffuse large B-cell lymphoma. IPI is the International Prognostic Index, basically says we're not treating patients who have stage 1 very low risk disease, meaning in just in one place. We're taking patients who are a little bit higher risk and treating them either with pola-R-CHP or R-CHOP. For 6 cycles, plus patients got 2 additional cycles of rituximab. And there were about almost 800 patients in this study. And the patients have been followed now for about 5 years. And what this study showed that, as with what had been seen in the original presentation back in 2021, was that there is a modest benefit in improvement in what we call progression-free survival, or how many patients stay in remission, if you add polatuzumab to R-CHOP chemotherapy. And this benefit is about 5 to 6%. Fortunately, when you add polatuzumab to R-CHP and you leave out the vincristine, the toxicity profile of these two drugs, these two regimens rather, is very, very similar. And there's a fair body of data out there that tells us there are subtypes within diffuse large B-cell lymphoma, what we call germinal center, non-germinal center. There's a lot of work to sort of subclassify within these diseases. There is significant data to support that patients who have the non-germinal center subtype of diffuse large B-cell lymphoma probably derive a bigger benefit from the inclusion of polatuzumab in their upfront chemotherapy. But this basically tells us that pola-R-CHP remains the standard initial treatment in diffuse large B-cell lymphoma. So what is a bispecific antibody? This is a new treatment that we now are getting a very good experience with in both aggressive and indolent lymphomas. And these are really kind of cool drugs. So what they do, and these are immunotherapy, they on one end bind to CD20, which is on the surface of nearly all B-cell lymphomas, at least when they initially present. And on the other end, they bind to your T cells, which are one of your own immune cells, which are responsible for helping destroy invaders into your body like bacteria or other things. And here you're bringing that T cell to the B cell and hopefully causing the B cells to be eradicated. And in some ways, this is similar to CAR T cell treatment, but this is what we call an off-the-shelf treatment. So we could administer it right away to any patient. We don't have to take their T cells and manipulate them. So this has really been a game-changing class of drugs in lymphoma. So this study from our friends in Australia called the Coalition Study compared patients getting polatuzumab plus R-CHP, which I just showed you, with the addition of glofitamab, which is a bispecific antibody targeting CD20, or glofitamab plus R-CHOP. And this was in younger patients who had diffuse large B-cell lymphoma. So this in some ways is similar to the prior study I showed you, though much, much smaller. There were 40 patients in each arm. And the idea here was to take higher risk patients, newly diagnosed, and give them one of these two regimens. Patients started out with R-CHOP, and then they were either randomized to get R-CHOP plus glofitamab or pola R-CHP plus glofitamab. And you can see here they're only getting, you start with a low dose of glofitamab and then go to the full dose. There were 80 patients have completed this study with a median, so patients are now approaching the 2-year mark. And what they found in this study was that patients did very, very well. And if you added glofitamab, this bispecific antibody, to standard treatment in previously untreated patients, you could give the full dose of chemotherapy. And that's really important because sometimes if you add something to standard chemotherapy, you actually have to dose reduce the chemotherapy because of side effects, and this can interfere with how well the treatment works. So 98%, nearly all patients had a complete remission with this approach. And at 24 months, a little over 85% of patients remained in remission. So this looks really exciting. It's a small-ish study. So now there are randomized studies ongoing, adding these bispecific antibodies to standard chemotherapy versus standard chemotherapy alone. And I think this is likely going to be our next approach in previously untreated diffuse large B-cell lymphoma. So we also use these bispecific antibodies in patients who have had prior treatment. And initially, these agents were looked at in patients who mostly had prior CAR T-cell treatment or had multiple other prior treatments. And when we do clinical trials, we often select patients, these patients have to get to an academic medical center. We have criteria in the clinical trial that we have to follow in order to say someone's a good candidate for a clinical trial. And sometimes these are very stringent. So we like to look after a trial has been performed and a particular drug is available to see how that drug actually performs in patients who are not being treated in a clinical trial. So these may be patients who are older or who have other medical issues who wouldn't have been eligible for the trial. And here they looked at both epcoritamab and glofitamab. Both of these drugs are approved in patients with relapsed and refractory diffuse large B-cell lymphoma. And what they saw in this analysis is that 70% of the patients in this analysis would not have been eligible for the clinical trial. And they saw that the response rates of both of these two bispecific antibodies looked quite good, but were a little bit lower than in clinical trials, which is what we typically see in this setting. But the outcomes between the two were quite similar. TP53 mutations are a marker of aggressive disease really across all cancers. And we know that in prior CAR T-cell treatment could theoretically make these drugs work less well, but they didn't actually see that. The patients still responded, though the response rates did differ whether you had a good remission after CAR T or a shorter duration of remission. And they also point out that when you give these agents, you want to make sure that there is CD20 on the surface of the tumor cells, because obviously that is the target of these agents. And for patients who've had a lot of prior rituximab, sometimes you can lose this, this marker on the surface of the tumor cell. Just wanted to include one CAR T-cell abstract here. This is raptacaptogene autoleucel. So this was a phase 2 trial. So all the patients got the same treatment. And this is targeting CD19 here, not CD20. And these are, we collect the T cells, we send them off, they're manufactured so that the T cells then recognize this protein on the surface of the tumor cells. We give some chemotherapy before we give what we call lymphodepleting chemotherapy before patients receive their treatment, and then we assess for response. And this, this is a newer CAR T cell therapy, and we're hoping that this could actually be manufactured more quickly. And what they found in this study was that this looked very active. The 88, nearly 90% of patients responded, and about 2/3 of patients had a complete remission. And 1 month after treatment, about 50% of patients remained in remission, and about 80% of those who had had a complete remission to the initial treatment. So this compares favorably to the data that's out there on CAR T cell treatment. Again, this is a phase 2 study, so you really need to do a randomized study to confirm that this is not just about the particulars of the patients they've selected for this trial. The other thing that was really encouraging about this particular agent was the safety. So things that we see in CAR T cell treatment typically are what we call cytokine release syndrome, which can be fevers and low blood pressure and need for oxygen and other organ toxicity, as well as neurologic toxicity, confusion, or other neurologic issues. And the rates of significant cytokine release or neurologic toxicity were very low with this particular agent, which is very exciting. So one other diffuse large B-cell lymphoma study I wanted to show was, CAR T-cell treatment has really been a total game changer for those of us who treat diffuse large B-cell lymphoma. But we know that not every patient can come to a CAR T-cell center. We know that some very elderly patients or frail patients may not be the best candidates for CAR T-cell treatment. So we need other treatments as well. And this was a study that took a bispecific antibody. I've mentioned glofitamab and epcoritamab. This is a third one called mosunetuzumab, which is given subcutaneously under the skin, and it was combined with polatuzumab. That's the drug I showed you in the POLARIX study. So those two were combined and compared to polatuzumab plus rituximab in patients with relapsed or refractory diffuse large B-cell lymphoma. And here is the study overview. This was a relatively small study, but patients were assigned to either get just polatuzumab rituximab or with mosunetuzumab. With these bispecific antibodies, you start with a low dose and then you increase the dose. And this was nice because it was time-limited treatment for 8 cycles of therapy. So what was found in this study was that giving the mosunetuzumab instead of rituximab combined with polatuzumab looked quite good with improved overall and complete response rates. The mosunetuzumab is, in terms of the bispecific antibodies, has a good safety profile and has fewer patients experiencing fevers or low blood pressure. And it is given subcutaneously, and this is a regimen that could be given in the community, in community practices, therefore reaching more patients. So there is an ongoing phase 3 study comparing these two, and hopefully this will become FDA approved and something that we can use in clinical practice. So when we look at all of the abstracts I've shown you in diffuse large B-cell lymphoma, some of the takeaway points are polatuzumab plus R-CHP remains the standard treatment for patients who have higher risk diffuse large B-cell lymphoma that has not been previously treated. The bispecific antibodies, we have epcoritamab, glofitamab, and mosunetuzumab are active in relapsed and refractory setting, and also looking very promising combined with chemoimmunotherapy in previously untreated patients. And CAR T-cell therapy continues to be our standard for patients who are eligible, who either have disease that doesn't go in remission after initial treatment or relapses within the first year, or in later lines of treatment. And these new constructs, these new ways of making CAR T cells are promising with faster manufacturing and lower toxicity rates. So switching gears to follicular lymphoma, this is the second most common subtype of non-Hodgkin lymphoma that we see, about 20% of cases. Again, a somewhat older age at presentation, a median of 60 years, with a slightly higher incidence in women. The reason it's called follicular lymphoma is when you look under the microscope, in a normal lymph node where your immune cells are doing their jobs, you would see these rounded areas or follicles. When you look under the microscope in many patients with follicular lymphoma, you see these rounded areas. That's why it's called follicular lymphoma. So when we first meet a patient with a slow-growing B-cell lymphoma, follicular lymphoma or a marginal zone lymphoma, we do staging as we do in every cancer. And if the disease is localized in one area and not elsewhere, we will often treat patients with radiation treatment. And this can result in long-term control of the disease. For patients who have more extensive disease, we know that treating right away versus treating when patients need treatment are so-called active surveillance patients. There's no evidence that there's a benefit to treating earlier. For people who have symptoms or have a higher burden of disease, we would typically give systemic treatment. So this was a study in patients who were newly diagnosed coming out of Memorial Sloan Kettering looking at mosunetuzumab. So that's that bispecific antibody. And in this study, it was given, as you can see here, subcutaneously. And in the prior study in follicular lymphoma, for which was approved by the FDA, this drug was given intravenously. So this is a nice option to give it subcutaneously. And patients would receive 8 cycles of treatment. Again, you start at a low dose and then go to a higher dose to lower the rates of having fevers or any sort of cytokine release syndrome. Patients were in complete remission after 8 cycles. They could then go on observation. If they had a partial remission, then they would continue on treatment. This is exactly how we use this drug in the relapse setting. And we're just basically now testing it in previously untreated patients. Things that you should know about the bispecific antibodies is the major other toxicity we see with these agents is risk of infection. So we put patients on preventative antivirals and antibacterials and follow people closely for risk of infection. So this data looked really very impressive. 96% of patients responded and 80% of patients had a complete remission with 1 year of follow-up. This is a short follow-up. More than 90% of patients remained in remission, which I think is really quite impressive. And giving this drug, particularly subcutaneously, was very well tolerated. So this is something that can be easily done in the clinic. And hopefully will be exportable to not just the academic centers, but to the community sites in the future. So this study was what we call a plenary. So with all of the hundreds of abstracts that are presented at a huge meeting like ASH, they pick the top ones for discussion at the plenary. And there were actually 2 lymphoma plenaries out of 6 talks, which is pretty cool. Not the typical thing because this meeting includes leukemia and benign hematology, sickle cell disease, lots of different topics. So it's really quite notable to have a plenary. So this study combined the CD19 antibody tafasitamab plus lenalidomide and rituximab in relapse and refractory follicular lymphoma and compared it to lenalidomide plus rituximab alone. And lenalidomide plus rituximab is a very commonly used study for patients who have had 1 prior line of treatment and need to be retreated with follicular lymphoma. And you can see here the design. Patients were treated with tafasitamab for 12 cycles, initially weekly, and then every other week. The lenalidomide was for a year, and the rituximab was given for 5 cycles. In the control arm, they actually gave an intravenous placebo, so nobody knew whether the patients, the treating physicians, no one knew who was getting what. And the primary, what they were looking at here was how many patients were progression-free over time. So in this study, the study met its primary endpoint, and there were more patients who progressed in the lenalidomide plus rituximab arm compared to the tafasitamab arm. And there was a 57% reduction in the risk of progression, relapsed lymphoma, or death. And this was seen across all subgroups. We know that when patients relapse with follicular lymphoma, if you relapse within 2 years of starting your initial therapy, the disease may be more aggressive. We call that POD24. And for patients who had not responded to prior rituximab or had had multiple lines of therapy, all of the patients seem to have, in all of those subgroups rather, patients did better with tafasitamab. We don't have overall survival data. We have such good additional options for patients who progress that we often don't see an overall survival benefit. And the safety profile looked basically what we would expect. And again, this is another well-tolerated, relatively easy to administer option that could be given at academic centers or in the community. This was another study in relapsed and refractory follicular lymphoma with a antibody drug conjugate called loncastuximab, which targets CD19 and delivers a chemo drug into the tumor cell in combination with rituximab. Loncastuximab is not approved in follicular lymphoma. It's being used in diffuse large B-cell lymphoma in patients who have relapsed or refractory disease. So this was a small study basically to say, well, we have this good drug in fast-growing lymphomas. How's it going to work in follicular lymphoma? And this schedule is relatively easy. So people come in 1 day every 3 weeks and get the combination of loncastuximab and rituximab. And then patients were assessed for response. And then if they were responding, could go on to additional cycles of loncastuximab and be treated for here initially 12 weeks, then 9 weeks, and then could get maintenance rituximab. So you can give additional doses of rituximab or additional doses of loncastuximab if not in remission. And the primary endpoint in this study was to, at 12 weeks, to see that more than half of the patients had had a complete remission. These were patients who had been pretty heavily pretreated. And again, there were patients who had progressed early and had high tumor burden. So this looked very good. And at 12 months, nearly all of the patients who had achieved a complete remission remained in remission. So that is an important number to look at. And this is a safe combination. The things that we see with this drug, patients can get some fluid retention and rash are the most common things that we see. But this was a tolerable regimen and something that, again, could be widely used if it was eventually approved for use. Switching gears, so this is a bispecific antibody, AZD0486. You'll notice that all of these drugs that are in clinical trials and not yet approved, they get these numbers before they, when they're initially being studied up front. And this testing in a phase 1 study, increasing doses of a CD19 instead of CD20 bispecific antibody. So it binds to CD19 and CD3. And this was in patients with follicular lymphoma. And patients, again, started at a low dose, and then you go up on the dose to try to, excuse me, prevent fevers or cytokine release. And you can see patients could get up to 2 years of treatment. This agent looked very active, overall response rate of 95% with a complete remission of 85%. These were patients who were heavily pretreated. And for those patients who achieved a complete remission, we can look in the blood to see, is there any trace of residual lymphoma? And in 92% of patients at 12 weeks, we could not detect any minimal residual disease. And again, this was quite well tolerated with mild cytokine release and neurologic toxicity. So the conclusions from the studies that I presented here in follicular lymphoma show that these subcutaneous bispecific antibodies alone and in combination are very active. This should say monoclonal antibodies, not subcutaneous, in previously untreated patients. Adding tafasitamab to lenalidomide plus rituximab looks very promising compared to lenalidomide plus rituximab. And again, this is a randomized study. This is the sort of most definitive of the studies that we use. So more to come on this, but this may be pretty rapidly taken up as a treatment option. And the bispecific antibody targeting CD19 that I just showed you is looking very promising in early data. So moving on to mantle cell lymphoma, this is what the disease looks like under the microscope. And when we think about patients with mantle cell lymphoma, previously, and this is beginning to change because of some of the data I'm going to show you, we think about, are patients younger and are they eligible to do something very aggressive like a stem cell transplant using their own stem cells in, assuming they achieve a remission with their initial treatment. That is one approach. We also have some data that I'll show you was updated from Europe, adding a BTK inhibitor that looked good. For older patients or those who are really not eligible for a stem cell transplant or those who have what we call TP53 mutations, we don't generally do high-dose chemotherapy approaches. And here the standard is typically to give bendamustine plus rituximab, followed by maintenance rituximab or a BTK inhibitor with or without a CD20 antibody like rituximab. So this first study was a very important study and was again a plenary study presented by Tim Fensky. So this was a study done across the US that asked the question, if you take patients with mantle cell lymphoma and you treat them with standard initial treatment, these are patients that you would normally take to a stem cell transplant because that has been our standard of care for many of years in the US. If you can prove that there is no minimal residual disease after initial induction and the patients are in complete remission by imaging and by bone marrow, patients were either assigned to get an autologous stem cell transplant with rituximab maintenance versus rituximab maintenance alone. For patients where they could not assess whether there was minimal residual disease, they went on to transplant, or patients who did not achieve a complete remission went on to transplant, both with maintenance rituximab. So this study actually showed for the patients who were in complete remission with undetectable minimal residual disease, whether you had a stem cell transplant plus rituximab or rituximab alone, there was no difference in progression-free survival. So this is going to be, I think, really put the nail in the coffin of taking patients to stem cell transplant in first remission with mantle cell lymphoma. So this has huge implications for how we treat patients, and I think is a big move forward because stem cell transplant obviously is an intensive treatment. Patients are in the hospital for a number of weeks and there are significant toxicities. What they found for those patients who still had minimal residual disease after their initial induction, there may be a benefit to stem cell transplant, but we need more data there. And some of those patients would then convert to being negative after stem cell transplant, and they seem to do quite well. I will note that the vast majority of patients after initial treatment with chemoimmunotherapy and with mantle cell lymphoma have undetectable minimal residual disease, at least 85%. So this was an update of a study that was presented last year at ASH, looking at adding ibrutinib to the initial treatment of patients who are younger, who are eligible for stem cell transplant. This is called the Triangle study, and it's a bit complicated. But basically, they took patients who were eligible for a stem cell transplant, and the standard of care in Germany would be to get chemotherapy, stem cell transplant, and then observation, or a little over half of patients got maintenance rituximab because as the study was being done, that became the standard treatment. And then there were two experimental arms. One added ibrutinib to this treatment. The ibrutinib was given with R-CHOP. You can't really give it safely with this RDHAP arm. And then patients after stem cell transplant would get ibrutinib with rituximab or observation, depending on when they were treated. And then the other arm was, well, what if you dropped the stem cell transplant altogether? And the conclusions from this trial is if you add ibrutinib to standard treatment with a stem cell transplant, those patients did better. If you took patients who got ibrutinib and did not have a transplant, doing the transplant was not better than that. And if you had a transplant with ibrutinib or the ibrutinib arm, there was really no difference in those arms. So that was what they had presented before, and really led to our thinking that, maybe we really shouldn't be doing stem cell transplant. And then along came the US study I just showed you, which really then confirmed that. They did see a better overall survival of the patients who got ibrutinib. So this really is, their conclusion was that patients should be getting ibrutinib upfront. They showed some data looking at higher risk patients who had what we call blastoid variant or proliferative disease, and concluded that those patients should get stem cell transplant, but that was very controversial and not statistically significant. So I think most of us didn't walk away with that conclusion, but we walked away with the conclusion that we should probably be adding a BTK inhibitor to the previously untreated population of mantle cell lymphoma patients, and not do a stem cell transplant and follow it up with maintenance rituximab. So that was interesting to see. So this study was very interesting. It was a phase 2/3. So they started with a phase 2 part that I'll show you here, comparing patients getting ibrutinib plus rituximab versus chemoimmunotherapy for previously untreated mantle cell lymphoma. And you can see the investigator had to decide before the patient went on the trial whether they were going to give R-CHOP chemotherapy or bendamustine plus rituximab. And then they were randomly assigned to ibrutinib with Rituxan versus Rituxan plus chemo. They got maintenance and then follow-up. And what was seen in this study was that the ibrutinib and rituximab arm looked better than the standard chemotherapy arm. But when they drilled down and looked according to what kind of chemotherapy the patient got, the improvement was completely explained by the R-CHOP arm. So ibrutinib plus Rituxan was significantly better than R-CHOP, but looked very similar to bendamustine plus rituximab. They also saw that for patients who had lymphomas that had that high proliferation index, that ibrutinib plus rituximab was probably less effective in that patient population. So, what do we do with this information? I think it tells, I think for most of us sort of walked away thinking, well, bendamustine plus rituximab probably is a very standard regimen for an older patient who would not tolerate a more intensive chemotherapy regimen, and then would save the BTK inhibitor for later because we're not seeing that it is superior, and you don't want to use up all of your good treatments early. But we'll need more follow-up on this. And again, this was with ibrutinib. We're not using ibrutinib in the US anymore because of toxicity when it's combined with chemotherapy. So more to come on that. And then lastly, switching gears to CLL, SLL. Here we can see an image of CLL in the peripheral blood. So for patients who do not have a TP53 mutation, options for initial treatment in the US tend to be venetoclax plus obinutuzumab, time-limited treatment versus a BTK inhibitor with or without obinutuzumab. For patients who are TP53 aberrant, we generally would use a indefinite BTK inhibitor with or without obinutuzumab. You could consider venetoclax plus obinutuzumab and continue the venetoclax. This is a bit more controversial. So this study, which was presented by Jennifer Brown from my institution, was a big randomized phase 3 trial in patients who were previously untreated, almost 900 patients, and they were randomly assigned to get acalabrutinib plus venetoclax for 14 cycles with or without obinutuzumab versus standard chemotherapy, which we really don't do anymore, but it takes time to accrue and run these trials, and that was the way that this was designed. And what they found was that by giving acalabrutinib and venetoclax with or without obinutuzumab, time limited, there was a very significant improvement in progression-free survival, meaning how many patients' disease was under control at 3 years. 83% with acalabrutinib and venetoclax, 77% with obinutuzumab, compared to 67% with chemoimmunotherapy. And we've seen many other trials looking at chemoimmunotherapy in CLL, and we really aren't using that approach anymore. But this was really good data to suggest that time-limited treatment incorporating these two drugs may be a very good option. They also looked at could they detect any minimal residual disease in the treated patients, and it was undetectable in the largest number of patients in the AVO arm. They didn't see a lot of cardiac side effects from the acalabrutinib. There were more infections and more toxicity adding obinutuzumab to the acalabrutinib and venetoclax arms, but this study was conducted during the pandemic, and we know that obinutuzumab can be a significant problem for patients who have COVID. I just want to mention briefly, this is so for patients with CLL who have had a BTK inhibitor like acalabrutinib or zanubrutinib, and then progress and may be treated with a drug called pirtobrutinib, then that is a group of patients that we are trying to develop newer, better treatments for. And there is a new class of drugs called Bruton tyrosine kinase or BTK degraders. And these are really very, very interesting drugs. So what they do is that they bind to the protein that you want to interfere with, and then they bind to what's called an E3 ligase. And what this causes is for these what's called polyubiquitination. So these moieties are put onto the BTK. So you can see this ubiquitination. And when that happens, these proteins, the BTK, is targeted for being broken down by what we call the proteasome. So this was a phase 1/2 study. So they're trying to figure out what's the right dose in patients with B-cell malignancies, multiple B-cell malignancies. But here, when we look at the CLL, SLL patients, we see that this is an oral drug. It was safe and well tolerated. And overall, the overall response rates in these patients who had had a lot of prior treatment was almost 80% in all patients and was 94% in the patients who had received 200 milligrams, which is, we think, the right, effective dose. We also saw some activity in patients who have Richter's transformation. This is when CLL becomes a more aggressive disease and can be very difficult to treat. There is now a phase 2 study looking at the BTK degrader in patients who have been previously treated with both a covalent BTK inhibitor, that's acalabrutinib or zanubrutinib, and pirtobrutinib. And then lastly, just wanted to present quickly epcoritamab in patients with relapsed or refractory CLL. So we've always wondered how these bispecific antibodies would behave and work in patients with chronic lymphocytic leukemia, because we know the T cells in patients with CLL may not function completely normally. So this was a phase 1 study where, an expansion study and optimization study, not a phase 1, but they start at a low dose, as we do with all of these bispecific antibodies, and go up on the dose. So this was the expansion cohort. And then because when you do this step-up dosing, you can still see a lot of cytokine release, so fevers and low blood pressure and other toxicities, they added an additional dose here, a 3 milligram dose. We use steroids and lots of hydration in patients getting these bispecific antibodies. And what they found was that these, again, are patients who've had loads of prior treatment. Nearly 40% of patients had a complete remission to epcoritamab, which was really pretty exciting. And in those patients who responded, again, this minimal residual disease was undetectable in 3/4 of patients. And by adding that additional 3 milligram dose, they were really able to lower the rates of cytokine release syndrome. So for the CLL, SLL, time-limited therapy with a BTK inhibitor and a BCL2 inhibitor looked very good with nice duration of remission. There are other combinations like this, that studies that are ongoing with BTK and BCL2 inhibitors, but I think this may be where we're moving to really try to use time-limited therapy in as many patients as we can. The BTK degraders, which is just such a very cool class of drug where you can get rid of any protein that you want to target, looked very promising in patients who are heavily pretreated. And the bispecific antibodies also looked very good in CLL. So I suspect we'll be seeing combinations in the future. So with that, I will stop sharing my slides and we can take
Thank you so much, Dr. Lacaze, for your excellent presentation. We're going to use the rest of the time we have, take a few minutes here to get to some questions. We're going to get into kind of some questions regarding some of the specific therapeutics and subtypes discussed today, but I did want to start off with a question about kind of the beginning portion of your presentation. So from start to finish, how long does it typically take for a new treatment to go from trials to approval? And how do you access phase 4 data?
It takes years to go from the preclinical data to developing a drug. When you do a phase 1 study, there is so much work that has to be done, and these are done very carefully with oversight by the FDA and others to make sure we understand the toxicity profile. And then you do a phase 1, and then it can be a phase 1 and 2 together, and then a phase 3. But it takes, many years, because you also have to follow patients who've been treated on these trials for a period of time before you can assess what the endpoints are of the trial to say whether it's a successful trial. And if you're looking at things like progression-free survival, that means lack of progression of disease. Generally we look at that at a couple of years out from treatment. So it really is a very, very long process. The phase 4 data, it's not generally, this is sort of collected all the time by the FDA, and sometimes they will, there will be alerts that come out when they look at toxicity that sort of pops up that hadn't been previously detected or hadn't been a concern. Phase 4, I don't think we do a lot of phase 4 trials in oncology, but in, diseases like cardiovascular disease or other where there are loads and loads of patients being treated, they do more phase 4 trials.
Thank you. Moving into DLBCL, can a DLBCL relapse patient be treated with glofitamab without chemotherapy?
Yes, the glofitamab was initially approved in patients who'd had at least 2 prior lines of treatment as a single agent, so it doesn't have to be given with chemotherapy. There is the STARGLO regimen, which combines glofitamab with gemcitabine and oxaliplatin, which was compared to gemcitabine and oxaliplatin with rituximab, and is now showed a significant benefit. That we are using. How much the chemotherapy adds to the bispecific, we don't really know, because they didn't randomize patients to glofitamab, GemOx versus glofitamab alone. So, we often try to tailor our treatment to the individual patient, depending upon patients, what patients want to do, and toxicity, and lots of other, it's an individual decision.
And how do you compare Pola-R-CHP to dose-adjusted EPOCH-R for DLBCL treatment?
So we don't really use dose-adjusted R-EPOCH for standard diffuse large B-cell lymphoma, because there was a randomized study done a number of years ago that showed no benefit to dose-adjusted R-EPOCH over R-CHOP. There were potential issues with that trial. It was a large national trial, and all of our clinical trials in diffuse large B-cell lymphoma tend to enroll healthier patients. If you're really sick and you're in the hospital, it's hard to get those patients onto a clinical trial. So these were a lower risk population of patients who didn't seem to benefit from the intensification of the dose-adjusted R-EPOCH. We do use dose-adjusted R-EPOCH in double hit lymphoma. That is when patients have a MYC and a BCL2 rearrangements. We don't have randomized data compared to R-CHOP in that setting, but the data that we do have in patients who are treated off of clinical trials shows that R-CHOP probably is inadequate. And we use dose-adjusted R-EPOCH in a rare subtype of aggressive lymphoma called primary mediastinal large B-cell lymphoma, where the dose intensity does seem to make a difference. So yeah, so it's very hard to know how those, we don't have a randomized trial with that, but it's really not a regimen that we generally reach for in most of our patients with aggressive B-cell lymphoma.
And for follicular lymphoma, what do you consider a high tumor burden?
So in most of these trials, they use the Guelph criteria. And that is a way, so if you're doing clinical trials in follicular lymphoma, because there can be incredibly variable amounts of disease, most of the trials have very strict criteria so that you can assess. So you're comparing apples to apples. Because if you treat patients who have, small burden disease with one treatment, you may see very, very good responses. So the Guelph criteria say 3 lymph nodes that are greater than 3 centimeters or 1 lymph node greater than 7 centimeters. That's one way we look at it. We can look at the spleen size and the blood counts, but I think that's one. 3 greater than 3 centimeters or 7 centimeters is often defined as high tumor burden, and that the study I showed you, the tafasitamab, lenalidomide plus rituximab versus lenalidomide plus rituximab alone, did require that patients meet the Guelph criteria.
You and for mantle cell lymphoma, will ibrutinib now be added to CAR T by default since it's shown to be beneficial during the trial? And if yes, when is that to be expected?
So CAR T cell, the trial looked at autologous stem cell transplant, not CAR T cell trial, and we really don't use ibrutinib anymore in general in the US because it was pulled from the market for the indication of mantle cell lymphoma because of toxicity based on a study where they compared bendamustine plus rituximab to bendamustine plus rituximab plus ibrutinib. So I think maybe what you're asking is, will we be adding a BTK inhibitor to initial treatment? And yes, I think we will. The challenging thing about that in untreated mantle cell lymphoma patients is the regimen that was used in that triangle study was 3 cycles of R-CHOP followed by 3 cycles of Rituxan plus Ara-C based treatment, which is not typically used so much in the US. So to apply that to US populations, we would typically think about using acalabrutinib or zanubrutinib, which wasn't studied in the trial. And, can we combine it with treatment regimens that are used more commonly in the US, like bendamustine plus rituximab, followed by rituximab plus cytarabine? I think that's all still a source of great debate in the field of mantle cell lymphoma. I think right now that's one of our most challenging questions. You meet a new patient with mantle cell lymphoma who is under 70 and trying to figure out how should we approach that patient. There are a lot of different options, and they're good options. So it's a good problem to have. But I think the bottom line is we probably will not be routinely taking patients to stem cell transplantation any longer.
Just wanted to touch on a CLL question here. As a high-risk CLL patient who was successfully treated in 2018 for Richter's transformation, I'm wondering if there are any promising trials or treatments in the pipeline for Richter's.
So Richter's is challenging. I think we're looking at there are some patients who've been treated with CAR T cells, but the problem with Richter's is it's very heterogeneous. There are some patients who have a disease that has never been treated before for underlying CLL. There's also a distinction of whether it is genetically related to the underlying CLL. And we know many of the cases are, and those are also harder to treat. So there's a big variety of aggressivity of Richter's. So it's really an area of unmet need. I think we're looking at the bispecific antibodies. There is a trial that we're participating in with a bispecific antibody, and maybe optimizing CAR T-cell treatment may be an option. But it is an area of significant unmet need.
And ending with a final question here, kind of speaking of bispecifics as an option, this person is wondering, when will bispecific antibodies be available at typical local infusion centers?
It's a great question. I think we're all waiting for that. And the Lymphoma Research Foundation was part of putting together a group of people to publish sort of guidelines on how to manage bispecific antibodies in community settings. So we are really trying to partner with our community oncologists to try to get them comfortable giving them, because we recognize that for many patients, it's just not feasible to come to, travel hours to start bispecific antibodies. What we do now is they'll often get the step-up dosing at the academic center and then go back to the community site. But I think with good resources and education, it really should be feasible to get these into community practices. And for patients out there, I would say, who are looking to get this, ask about it a lot, because I really do think this should be able, and particularly now that we have some subcutaneous appears to have less toxicity, adding additional step-up dosing. There are strategies that have been developed that really reduce the risk of cytokine release syndrome.
Thank you. I think that's a promising note to end on. I want to thank everyone for joining today's webinar. I know we got a lot of questions, so please always remember, if you have additional questions after today's webinar, you can always reach out to our helpline at 800-500-9976. And you can find our number and our email for our helpline at lymphoma.org always. But with that, thank you so much, Dr. Lacaze, for your time today.
Right, great. Thanks for
Thank you,
joining.
everyone.
Bye.
Take care.